1. Test result. One analytical method, and a single wrong decision can shut down production, trigger recalls, or invalidate years of data. Analytical method validation isn't just a laboratory requirement. It is a legal, scientific, and regulatory necessity. In this video, you'll understand why validated methods are critical, what regulators expect, and which validation parameters truly matter in real-world applications. So, what is analytical method validation? It's the documented process that proves an analytical method is suitable for its intended purpose. In simple terms, it answers a critical question: can we trust the results this method generates? Validation ensures that laboratory data is accurate, reliable, consistent, and scientifically defensible. Think about it. Validated methods. Your test results are just assumptions. Assumptions are completely unacceptable in regulated industries. You need to be sure your method is fit for purpose, delivering reliable results backed by solid scientific evidence. Why are validated methods so important? In regulated industries, analytical data drives critical decisions. Do we release or reject this batch? Do we approve this new supplier? Does our product meet the required specifications? If the method used to generate that data isn't validated, every single decision based on it is at risk. Regulatory agencies don't evaluate opinions, they evaluate data. And data is only acceptable when it's generated by validated methods. Sure batch release, your regulatory submissions, and your entire compliance framework depend on it. In the United States and globally, method validation is not optional. It's a mandatory obligation. Regulatory bodies like the FDA require that analytical methods be properly validated according to recognized standards. Key guidelines from organizations like the USP, ISO, and the ICH define exactly how this validation must be performed and documented. During inspections, auditors will absolutely review your validation protocols, your results, and your final conclusions. If your methods aren't validated or are poorly validated, you can expect observations, warning letters, and even more severe enforcement actions. A critical concept in method validation is fit for intended use. This means the scope of your validation must perfectly match how the method is actually used in the lab. For example, a simple screening method does need the same exhaustive level of validation as a quantitative method used for final product release. Over-validating wastes time, money, and resources. Under-validating creates massive regulatory risk. The key is to perform a risk-based validation that is appropriate for the method's specific purpose. Now let's dive into the main parameters of validation. First up is linearity. Linearity evaluates whether the method produces results that are directly proportional to the analyte concentration within a specific range. If you double the concentration, you should see a doubled response. Poor linearity completely compromises your ability to quantify accurately, leading to flawed decisions. Next, we have the limit of detection, or load, and the limit of quantification, or lock. The load is the lowest amount of an analyte that can be detected by the method. The lock is the lowest amount that can be reliably quantified with acceptable accuracy and precision. These parameters are absolutely critical when you're dealing with impurities, contaminants, or any kind of trace-level analysis. Then there's precision, which evaluates how close individual test results are to each other. We look at this in two ways: repeatability and reproducibility. Repeatability measures variation when the test is performed under the exact same conditions, same analyst, team instrument, same day. Reproducibility evaluates variation across different conditions, like different days, different analysts, or different instruments. Inconsistent results are a clear sign of a method that simply cannot be trusted. Accuracy measures how close your test result is to the true known value. It's important to remember that a method can be very precise, but not accurate. Spots. Imagine hitting the same spot on a target over and over again, but it's the wrong spot. That's a precise but inaccurate method. Accuracy confirms that your results reflect reality, not just consistency. Next are selectivity and specificity. These parameters assess the method's ability to measure the target analyte without interference from other components in the sample. This is especially important in complex samples like food, pharmaceuticals, and biological fluids, where many other substances could potentially skew the results. Finally, we have standard deviation and measurement uncertainty. Standard deviation gives you insight into the variability of your results. Measurement uncertainty quantifies the doubts surrounding a result, essentially giving you a confidence interval. Regulators are increasingly expecting laboratories to understand, calculate, and justify their measurement uncertainty, especially for critical tests that inform major decisions. It's crucial to understand that method validation is not a one-time activity. Any change to your equipment, reagents, analysts, or even a sample type might require revalidation, or at least a verification study. Laboratories must maintain the validated state of their methods through robust change control systems, ongoing performance monitoring, and periodic reviews. A method that was validated today is not guaranteed to be valid tomorrow without proper oversight. So, what are some of the most common errors we see in method validation? Failures often stem from incomplete protocols that don't define the plan clearly. Another major issue is poor statistical treatment of the data, leading to incorrect conclusions. Setting inadequate acceptance criteria is also a frequent problem. If your criteria are too loose, the validation doesn't prove anything. And, of course, a lack of thorough documentation is a guaranteed way to fail an audit. Regulators look beyond the numbers. They evaluate your scientific rationale, your risk assessment, and the overall integrity of your data. Analytical method validation is the absolute foundation of reliable data, regulatory compliance, and sound decision-making. In regulated industries, data integrity starts with validated methods. Thanks for watching. Subscribe to this channel for more practical insights on analytical validation, quality systems, and regulatory compliance.